This is a podcast from the Nuffield Department of Medicine. Professor Tomáš Hanke tells us about his research on HIV vaccine development.
Hello, Tomáš.
Hello.
HIV shows huge antigenic variation. How can we develop a vaccine?
That is correct. The biggest challenge in development of effective HIV vaccine is the enormous HIV variation, which means that HIV has enormous capacity to change. That is, every time body launches an immune response as a result of natural infection or after vaccination, HIV changes its code, becomes invisible to these immune responses and escapes. Fortunately, there is a limit to HIV variation, and even HIV cannot change completely and has to keep small parts of its proteins conserved so that it survives. And it is this Achilles heel of HIV that we are focusing on by our vaccines. This comes with another advantage, that is, on different continents or on different geographic regions, HIV evolve into different subtypes. But again, all these subtypes have to have these conserved regions in common, and therefore if our vaccine works, it can be deployed in Africa, Asia, Europe, America. It would be universal. Finally, the strength of our vaccine approach also comes from using a unique and very potent combination of different vaccine subtypes, which together induce very strong immune responses.
And how can we stop the transmission from mothers to their babies?
Most, but not all, HIV transmission from HIV-infected mothers to babies can be prevented by antiretroviral drugs. However, in developing countries, these drugs are not readily available, and their use is associated with harmful, unwanted side effects. And also there is a possibility that HIV again changes and becomes, in this case, resistant to HIV drugs. Therefore, as a result, an unacceptably high number of babies become infected through breastfeeding. Just like for the whole HIV epidemic, the best solution for protecting babies against HIV is development of a safe and effective HIV vaccine given to adults to decrease the number of HIV-infected mothers, and given to babies at birth to protect them against their mother's virus in the breast milk. Just like for the adult vaccine, we are developing a pediatric vaccine which focuses on the conserved regions of HIV.
And can your research help us to design other vaccines?
There are many difficult infections and diseases like cancer, which can use exactly the same tools to induce strong and protective immune responses. And just like we are learning from other groups and their approaches and innovations, other groups can learn from us.
And what are the most important lines of research that have developed over the past 5 or 10 years?
Many new exciting molecular tools and technologies have become available, which on one hand allow us to induce more potent and vigorous immune responses, and on the other hand allow us to measure them and characterize them in much more greater detail. As for HIV vaccines, after the first two failures of candidate HIV vaccines, the third vaccine in so-called Thai trial induced marginal protection, and this was an enormous boost to the whole HIV vaccine development field. Of course, this observation has to be reproduced.
So why does your line of research matter? Why should we put money into it?
HIV infection and AIDS remains one of the most pressing global health problems, and development of a safe, effective, accessible, preventive HIV vaccine is the best and cheapest solution.
And how does your research fit into translational medicine within the department?
Developing vaccines is an excellent example of translational medicine. While testing novel ideas for vaccines in models is important and saves lots of money and time, at the end of the day what matters is what happens in humans. Our group tests lots of ideas in the laboratory, but focuses on safe and rapid translation of the most promising observations into humans. And therefore our programs fits very well with the translational activities of the department.
Thank you, Tomáš.
Thank you.